Okay, when we ended up with the notes last time, we were talking about the structure of DNA. And one of the things Watson and Crick first thought was like all the nitrogen bases, they just paired with themselves, like A went with A, T went with T, and so on. But if you look at that, remember, you have purines and you have pyrimidines. You have single ring and you have double ring. Well, if you have two single rings together and then two double, you have a two pair and then a four pair. And if you look at the pictures, it was uniform throughout. There wasn't this in and out, in and out. So they knew that that could not be the case. There was no way that they paired. So then they started looking, okay, purine is one and pyrimidine is two. Let's look at those guys instead. And sure enough, they made it even because you'd have a one ring and a two ring. So all of the steps would actually be made out of three ringed, a purine and a pyrimidine. But again, they weren't exactly sure which ones. Then you come into, well, here's why it wouldn't work. Okay, if you put two purines together, it'd be four, and then you'd put two pyrimidines together, it'd only be two. So it would go like this, and that's not the way the X-ray data, that picture showed it. So they knew it had to be appearing like this because then it would always be three, and it would always be the same distance apart. Okay, so they weren't sure. They knew they had to be specific, but they just weren't sure which ones went together. They determined that adenine and thymine went together and guanine and cytosine. This wasn't because of their work. This is because of Chargaff's work. Remember, we talked about him. He's the one that figured out in living organisms, the amount of adenine and the amount of thymine are always the same, and the amount of guanine and cytosine are always the same. So they looked at that and went, hey, wait a minute. Okay, if these two always equal each other, they must be the two that pair together. These must be the ones paired together. So they needed his work to figure out which ones paired, because originally they just thought all the A's, the T's, they paired with themselves, but that wouldn't work because of their size. Okay, so now we know that adenine always goes with thymine and guanine always goes with cytosine. Okay, that's been proven over and over and over. And that's how the code's made too. Okay, so now when a cell's getting ready to divide, it needs to make copies of that. And the speed at which DNA is made is crazy. It's really fast and it's very, very accurate, which is really probably the most important thing. There's more than a dozen enzymes and other proteins that participate in it. There's a process. I'm not going to expect you to know all the proteins and everything. That'd be a little crazy. Okay, but when it's ready, okay, let's say the cell's getting ready to divide, so it needs to copy all of its DNA because when it divides, then each new cell has to have the same DNA. They have little sites that are called origins of replication, and they kind of open up, the strands start to separate, and they form what they call a bubble. And on a DNA strand, you might have more than one because when they start replicating both ways. So in a chromosome, in a eukaryotic chromosome, they can have hundreds or even thousands of origins of replication. It just depends on the organism. But because it has so many, it goes really fast. Okay, so they can make copies of DNA pretty quick. Okay, the replication from that bubble goes in both directions. So the entire molecule is copied. And it's not just from one end to the other end. It can go into parts in between. Here's an example of bubbles. Okay, and I have a video I'll show you after this where you can actually see the DNA replicating. Okay, again, what it does is you see there's all kinds of enzymes but basically here's helicase. It opens it up. It's like a zipper. So now what's going to happen is these guys have their parts hanging out. There's going to be nucleotides that come in here and make copies of it. Now here they have an example of when RNA comes in, because if RNA needs to copy this gene, it'll come in when it's open, make a copy of it, and then it'll leave. But the DNA, if it's making a copy of itself, it'll just, this side will add on and this side will add on. Okay, they'll, if this was AAC, then TTG would come in here and they would just keep going down the line and they would replicate it. Okay, so an enzyme that can start it is called primase. Okay, it could start an RNA chain from scratch as well. So in other words, when the DNA opens up, the RNA nucleotides can make a copy of it. And it works really slick. So you can, while you're copying DNA, you can also make, if the RNA actually needs to make a gene, to copy a gene to do something, it'll make a copy of it, and then it'll go out of the nucleus into the cytoplasm and start making the proteins. Okay, the primers to start it, the ones where the bubbles start, are pretty short, 5 to 10 nucleotides long. Okay. And they usually start on the 3. When you look at DNA, it goes from 3 to 5 or 5 to 3. They flip it over. It usually starts on the 3 end, not the 5 end. Okay. So we've got these enzymes. They're called DNA polymerases. And again, remember, enzymes usually end in ASE. But they're the ones that start this replication. And they make it, they spread it out, untwist it so that it can happen. They're the ones that basically change the shape of the DNA so you can open it up like a zipper and put in your new DNA, your new nucleotides. Okay? They usually require primer, in other words, that bubble. And then you have your DNA template stand. So each part of the DNA, when it gets split, is a new template. Okay? The rate at which this can happen, if you're talking of bacteria, 500 nucleotides per second. It zips. In human cells, it's more like 50 per second, but still, that's pretty quick if you think about it. That's how fast it can copy DNA. Okay? Each nucleotide that is added to the growing DNA makes it, bigger and bigger. It's a trinucleoside triphosphate. In other words, a nucleotide with your sugar and your phosphate and 1 nitrogen base. Okay? And it needs energy. In order to do this, it gets its ATP. They call it DATP. It's, it actually supplies some adenine to DNA because that's what's in ATP is adrenaline triphosphate. So you've got one of your nitrogen bases right there. Okay. The difference in the sugars, and again, we talked about this, deoxyribose is the sugar in DNA and ribose is the sugar in RNA. And if you look, deoxy, it just means that the sugar in DNA has one less oxygen than the one in RNA. But again, once it's open, it can copy both. Okay? So each monomer of it joins a DNA strand. So each one of those nucleotides goes in there and it just brings its sugar, its phosphate, and its nitrogen base, and it attaches, and then another one, and they just keep going down the line, and then they join together, and you got yourself 2 strands of DNA where you used to have 1. Okay? They actually have enzymes that proofread it. Okay, so if the nucleotides are not correct, they'll go in and fix it. So it's not just making the DNA, it's fixing it. If you have mismatched, if you have an A going with a C when it's not supposed to, it'll go in and fix that. So there's other enzymes that go in and proofread it and repair it if it's wrong. Okay, things that can damage DNA, if DNA is exposed to chemical and physical agents, cigarette smoke is a really bad one. X-rays, asbestos, because of those things, they can actually change the structure of DNA. And if that happens, you end up what they call a mutation. And that's not a good thing. We talked a little bit about that when we talk about chromosome inheritance. If you have things that pieces missing or whatever, that can happen because of exposure to harmful agents. Okay. They also have another repair. A nuclease cuts out and replaces. If you have a whole piece of a whole area that's damaged, because usually it's just off by a nucleotide or something. But if it's a whole piece, there are some mechanisms that will fix that. Okay. They'll cut out a whole piece and then it'll get replaced the right way. Okay. So the error rate in proofreading is low, but it's not perfect. So you're still going to sometimes end up with errors. And again, you're going to end up with you could end up with problems. Sometimes an altered DNA might lead to like a birthmark. Not a big deal. Okay. But there's, when you get altered ones, another one I forgot to mention is some drugs. You can read stories about women that were given drugs to stop miscarriages or something, but they ended up having children with no arms or legs. And, there's all kinds of stuff like that too that can cause problems for the DNA. Okay. Sequences changes can become permanent if it isn't detrimental. In other words, it doesn't cause the organism to die. It can be passed on the next generation. If, like I said, if it's not something that causes that individual organism to die. Okay. Those changes are called mutations, and they're a source of genetic variation. Okay. Sometimes a sequence change causes, like maybe it causes giraffes to be taller, their necks to be longer. Well, that's an advantage. They can get more food because they can reach higher leaves on trees. So it depends on. So again, mutations are changes. Some are good, some are bad. Okay. All right. There's limitations to the problems it can fix, okay? Sometimes the replicating ends don't stop when they're supposed to, things like that. It's not common, okay? The usual replication provides no way to complete the 5 to 3 end, so sometimes they have to repeat rounds of the shorter DNA molecules, okay? It depends on the DNA. It's not a problem for prokaryotes because they have circular chromosomes. They don't have X's like we do. So sometimes it has to be modified. But for a prokaryote, it's simple because it just goes around in the circle. All right. Eukaryotic chromosome DNA, okay, true cells, they have special nucleotide systems on the end. They're called telomeres. It's like you need to stop here, okay, because we had to start, we need to stop. So telomeres do not prevent the shortening of DNA molecules, but what they do is postpone, so ends don't break off. So telomeres is the end. This is where you quit, and they're tougher, so they make sure that the ends of the DNA molecules don't erode off. Okay? They say now that as you get older, the telomeres wear out, and then you get your DNA, parts breaking off, and then they don't work anymore, and they think that might be part of aging. Okay, so here's the telomeres. You can see it. They're on the very ends of chromosomes, and that's where you stop, and there's also supposed to kind of like protect the chromosome from eroding. Okay. In a chromosome, if germ cells are normal body cells. Okay. So if they become shorter every cell cycle, you're going to start losing some genes. So that's why you really need to make sure that doesn't happen. Okay. Eventually, you're going to end up with gametes that are totally missing genes, and then you're going to have some problems. Okay. So they actually have enzymes that catalyze the lengthening of those telomeres in germ cells. So in other words, if they start to look like they're eroding, they help get them back. They help elongate them back to the way they were. Okay. So when you're looking at bacterial chromosomes, it's double-stranded, but it's a circular. Remember, bacteria doesn't have a nucleus or organelles, but it does have DNA, and it's usually a circular strand, or circular double-stranded, but it's just one little circle, and that's their chromosomes. I guess you'd call them chromosomes. Okay. We have linear DNA. All eukaryotic chromosomes have linear DNA. So we have a lot more protein. But again, you think of a bacteria, one cell, really tiny, whatever. So their information, they don't need nearly the genetic material that we have. Okay. So in our bacteria, we call it supercoiled. And like I was saying before, it's long and skinny and stretched out. But when it's time for the cell to divide, it coils down into really, around proteins, and then that's why you can actually see it in a regular microscope. Okay, and the protein that it curls around is called chromatid. So a chromosome is made out of protein, chromatid, and DNA. And the chromatid causes the DNA to coil up and get to the point where it's small enough that it can be copied and then sent off to the new cell. Okay, chromosomes fit in the nucleus, and there's a big fancy packaging system. You don't have to know about it. Okay, here's what it looks like. If you look at a cell that's getting ready to divide, and this one's they've been stained, here's all your chromosomes. Okay, normally you can't see DNA. It's too skinny and stretched out. But when it's getting ready to divide, again, they coil up, and then this is what they call a karyotype. This would be lining them all up. Okay, and again, we have 22 pairs of autosomes or body chromosomes, and this person only has an X. Weird. So this person has, if you remember Turner syndrome, they're missing one of their chromosomes. I don't see the Y anywhere, and I don't see another X. Okay? So anyway, this is a karyotype. This is what your chromosomes would look like. And again, we are done with chapter 16.